Welcome, my dear students. In today's class, we explored the fascinating world of protein architecture, the tertiary and quaternary structures of proteins. These higher levels of protein organization determine the precise three-dimensional shape and biological function of every protein molecule inside living cells. We will understand how long-range interactions fold a polypeptide into a compact shape, how multiple subunits come together to form functional protein complexes, and how examples like myoglobin, hemoglobin, collagen, and enzymes beautifully demonstrate structure-function relationships. Let's unfold the elegance of protein folding in a simple and clear way. Let's begin. What is tertiary structure? Tertiary structure is basically the complete three-dimensional arrangement of all atoms in a single polypeptide chain. It represents how secondary structural elements such as alpha helix, beta sheets, turns or loops fold and they pack together. This three-dimensional shape determines biological function such as enzyme activity, ligand binding, etc. How tertiary structure differs from the secondary structure. Level of the first feature is the level of folding. So in secondary structure there are local folding, but in case of tertiary structure there is overall folding. Range of interactions. Secondary structures, in case of secondary structures, interactions are found between neighboring residues, and in case of tertiary structure they are found between distant residues. Examples of secondary structures include alpha helix, beta sheet, etc., and tertiary structure include complete 3D protein shape. Long range interactions. Amino acids that are far apart in the primary sequence may come close together after folding. Examples include in enzymes amino acids from different regions from the active site pocket. Role of the cellular environment. Folding often occurs with the help of chaperone proteins, and chaperones prevent misfolding and aggregation. Incorrect folding may lead to diseases like Alzheimer's, Parkinson's, and prion diseases. This is the image showing the tertiary structure of protein where different types of bonds are found, just like ionic bond, the covalent bonds, hydrogen bonds, etc. Importance of bend producing amino acids. There are certain amino acids cause bends or turns that help the protein fold properly. Proline is a rigid cyclic structure, introduces sharp bends. Glycine is very small, it fits into tight turns. Serine and threonine, their side chains allow flexibility and H bonding. These residues are often found in beta turns and loop regions, guiding the folding direction. Forces stabilizing tertiary structure. Tertiary structure is stabilized by multiple interactions such as hydrogen bonds. They are found between backbone atoms or between side chains. They help maintain helix sheet packing. Ionic bonds, they are located between positively and negatively charged side chains such as lysine, glutamate, etc. Hydrophobic interactions, they are found in the nonpolar amino acids like valine, leucine, isoleucine, phenylalanine, and they cluster inside the protein to avoid water. This is the primary driving force for folding. Van der Waals forces, weak attractions between atoms that are close together. They provide fine tuning of packing. Disulfide bonds, they formed between the two cysteine residues such as cysteine SS cysteine. Their strongest interactions, they stabilizes proteins exposed to Hertz conditions such as extracellular proteins like insulin. What is quaternary structure? When a protein has multiple polypeptide chains or subunits, their three-dimensional arrangement is the quaternary structures. And these subunits can be identical or known as homomeric, and they can be different, that is heteromeric. Such as example, hemoglobin has 4 subunits and DNA polymerase is a multi-subunit protein. So there is the primary amino acid sequence is the primary structure of protein, and it will form the local conformations. They are the secondary structure. In a polypeptide chain there are various secondary structures are present, but various secondary structures when make up the three-dimensional appearance of a single polypeptide chain, that is tertiary structure, and when within a protein there are multiple polypeptide chains and they interact with each other through different kinds of bonds, they're called as quaternary structures. So to conclude, tertiary and quaternary structures of protein give proteins their final functional form, allowing them to act as enzymes, structural elements, transporters and regulators of life processes. While tertiary structures describe the unique 3D folding of a single polypeptide chain, quaternary structure explains how multiple subunits unite to form powerful protein assemblies. The stabilizing forces, folding patterns, and real biological examples we learned today show how beautifully structure governs function in living organisms. Keep revising these concepts. They form the foundation for understanding biochemistry, molecular biology, and many competitive exams. See you in the next class. Thank you.